The mechanism of cell-mediated immune defense, where an effector cell of the immune system actively lyses a target cell whose membrane surface antigens have been bound by specific antibodies. The goal is to identify, target, and lyse cells to help contain pathogens. The lymphocytes in charge of this process are the CD-positive cytotoxic T cells, also known as CTLs. These cells help with the defense against cytosolic pathogens, which are mostly viruses. CTLs can also recognize and kill cancer cells. We're going to address the two processes in which CTLs are able to kill target cells. First, they release two types of cytotoxic proteins. Granzymes induce apoptosis in any type of target cell. However, they must be inside the cytosol of the cell in order to induce apoptosis of the target cell. The second protein, perforin, is a pore-forming protein. It punches holes in the target cell membrane, which allows the granzymes to enter. This process allows the CTLs to kill practically any type of virus in the cytosol of a cell. The other mechanism CTLs use to target pathogens is through FAS-L. Fas ligand is a membrane-bound molecule which is mostly expressed by CD8-positive T cells. However, some CD4-positive T cells also express this ligand. When it binds to Fas on the target cell, it induces an intracellular signaling cascade which leads to apoptosis of the target cell. It's important to remember that the T cells have undergone extensive training in order to recognize only the antigens present on the MHC class I molecules. So CTLs need to be activated by APCs in order to trigger this. Additionally, this process is very precise to minimize tissue damage while eliminating all of the infected cells. Apoptosis is the preferred method of killing pathogens because this process will activate nucleases, which will destroy cellular DNA and also degrade viral DNA. This is important because once the cells get destroyed, there will be no infectious virions that could infect nearby cells, whereas during necrosis, the intact pathogens are released from the dead cell and are able to infect nearby healthy cells. During lab, we use the splenocytes from both the naive and pathogen-inoculated mice. These will be known as the effector cells. The target cells will be the EL4 cells that were infected with H1N1 influenza virus. Both the target and effector cells will be mixed together in this experiment. The recognition of the MHC peptides on the target cells, which are the AL4 cells, by the effector cells, the CTLs, will lead to apoptosis of the target cells. In order to quantify the cellular cytotoxicity of this experiment, we conducted an LDH cytotoxic assay. Lactate dehydrogenase is a cytosolic enzyme that is a good indicator of cytotoxicity. When the plasma membrane of the cell is damaged, LDH is released into the surrounding cell media. Through an enzymatic reaction in which LDH catalyzes lactate to pyruvate via NAD+ reduction to NADH, a red formazine product is produced. The level of formazine formation is directly proportional to the amount of LDH released into the medium, which is indicative of cytotoxicity. The goal of this experiment is to use an LDH cytotoxicity assay to quantify cellular cytotoxicity by measuring formazine spectrophotometrically at 490 nm. In order to determine the percent cytotoxicity of the experiment, we needed to determine the LDH activity. This was done by subtracting the 680 nanometer absorbance value from the 490 nanometer absorbance, as seen by the formula on this page.
The table is separated into two separate sections, indicating the determined values at both 490 and 650 nanometers. At both of these absorbance values, an average was determined for the naive splenocytes, influenza splenocytes, negative control, and the maximum LDH control. Using the formula from the previous slide, the percent cytotoxicity was calculated. As a result, it was determined that our naive splenocyte presented with 11.4 toxicity in comparison to the inoculated H1N1 splenocytes, which we calculated to have a negative value, indicating that formazine was never produced during the LDH assay. This negative value indicates that we didn't have CTLs present. An explanation for these findings could be due to the fact that the mice inoculated with the virus were only exposed for 1 week. It is possible that a proper adaptive immune response wasn't formed, thus the lack of cytotoxicity.